Thank you. Can I get my slides up front? Today I will discuss with you the role of the gut-brain axis and nutrition, and how nutrition can affect gut-brain axis, and also give some more practical examples of that next. Next slide, please. To understand that the gut-brain axis and how it interacts with nutrition, we have to know a little bit more about the gut-brain axis. And what you see here is that I've depicted 5 of the most important parts of the gut-brain axis. The first is the intraluminal contents of the gut, which is mostly microbes, remnants of food which is not digested, and all sorts of antigens, toxicants, hopefully not too many, and so on. Virus also. The second part is the gut mucosa, and that's actually, I will come back to that on the function of the gut mucosa a little bit later in my talk as well, but it is the main barrier between the intraluminal content and the rest of the gut. And then a little bit below the mucosa, you have a lot of specialized cells, and not to forget also parts of what we call the enteric nervous system. So that's the gut's own nervous system, which interconnects with the brain, and that is the link to which you see down in the slide, but it also interconnects with the mucosa. So here we have a combination of specialized cells which can actually interact with the lumen also, and with bacteria, and interact with the nervous system as well. And then the connection between this enteric nervous system and the brain is mainly by the vagal nerve, and that enters the brain and the brainstem, as you can see in number 5. That's the neural route. The humoral route is just by taking up substances and compounds in the gut, entering the portal circulation, going to number 4, as you can see, and then metabolized in the liver. And if molecules are small enough to pass the blood-brain barrier, which you see at number 5 again, they can also enter the brain, and that is the humoral route for gut-brain interaction. If we look at brain-gut interaction, it's more or less similar. The neural route goes by the vagal system as well, vagal nerve, and the gut-brain on the humoral side goes actually mainly by hormones produced in the brain, like you can see in the hypothalamus, where corticotropin-releasing hormone is produced, which has a direct effect on what you can see left, the mast cells. So that is the humoral route on gut-brain interaction. So it's quite complicated, and next slide will give you a sort of overview. Next slide, please. An overview of the mediators which are involved in bidirectional signaling, because it's important to understand that it is not only gut-brain, it is also brain-gut interactions. And what you see here is just that there are numerous signaling compounds between the gut and the brain, and that makes it quite complicated. I will go into a little bit more detail on serotonin. Next slide, please. Which is one of the, I would say, more important signaling molecules between the gut and the brain, but also it has direct practical consequences, which I will describe a little bit later in my talk as well. So of serotonin, only 3% is in the brain, and most of the serotonin is actually in the gut. More than 95% is a little bit also in the thrombocytes. Important to understand is that serotonin as such can't pass the blood-brain barrier. However, tryptophan is the substrate of serotonin. Tryptophan is an essential amino acid, and tryptophan can pass the blood-brain barrier. And we know that the synthesis of serotonin out of tryptophan is heavily affected by, for example, inflammation and stress. And that's one of the reasons inflammation and stress has an effect on the brain by gut-brain interaction. And I also will show an example of that a little bit later in my talk. So this has really clinical consequences. A little bit more about the serotonin and experiments we did for some quite a while ago, but are classical examples and classical experiments now today. Next slide. And here you see gut-brain interaction again. Next. What we have done is that we have inflated a balloon in more or less healthy people as well as IBS subjects, inflated a balloon in the last part of the intestinal tract, the rectosigmoid. Next slide. If you do that and you measure actually brain activity using functional magnetic resonance imaging, you see certain parts of the brain stimulated and activated. The next slide shows you exactly the same experiment, but the difference is that it is also in exactly the same persons. But the difference is that we made these subjects for some hours actually very low in tryptophan levels and also very low in serotonin levels. So the only difference is the serotonergic signaling between the left and the right panel of this slide. And you see in the right panel that other parts of the brain were activated in exactly the same experiment. Next slide gives you also a more overview, and you have not to go too in detail. It is just showing how different parts of the brain interact with each other, either stimulate or depress each other. And you see that there is quite a different pattern in the right part of the panel compared to the left part of the panel. So that means that just only changing serotonergic signaling, it actually changes gut-brain signaling quite heavily. So this is more or less the proof of that serotonin is heavily involved in gut-brain signaling. Next slide. We have talked about serotonin. Now we shall also talk about the microbes, the gut microbes, because they are also important in understanding how gut-brain signaling works and how it interacts with nutrition. As you know, nutrition will also affect the gut flora, the microbiome in the gut. So here you see examples of how microbes directly can interact with the host, normally the gut mucosa. And it can be either activation of the vagal nerve or the enteric nervous system and so on. But that's what we call the direct microbe-host interaction. So the microbe directly interacts with a receptor on the host. The next slide shows you another way of how microbes can interact with gut-brain signaling, and that is by the substances they produce, the metabolites of these microbes. And one of these metabolites are the short-chain fatty acids, and they are extremely important. They are metabolites by carbohydrate fermentation by the microbes. And one of the most important one of these is butyrate, others are acetate, propionate, and so on. And especially butyrate has been subject to a lot of research and have shown to have a beneficial effect, not only on the gut itself, but also on the metabolic system, as well as also on mental health. I also mentioned isoforms of the fatty acids, like you can see in isobutyrate, for example. And these isoforms are normally the result of fermentation and degradation of proteins in the large bowel. And unfortunately, these isoforms normally compete with the short-chain fatty acids. So the isoforms are also called branched-chain fatty acids, and they compete with the short-chain fatty acids on the same receptor, although not effective. So they have actually a negative effect on butyrate signaling. Next slide. So we have talked about the microbes, we have talked about nutrition a little bit, and we go through the gut-brain axis and understand how the gut-brain axis works. And the last part of the gut-brain axis I want to explain to you is the intestinal barrier function. So I showed it already in the first slide, the mucosal part of the epithelium. Intestinal barrier function is extremely important because it is the barrier between the intraluminal part of the gut where all the nutrients are and the rest of the body itself. And like a spam filter in a computer, You want to get your emails, but you don't want to get your spam mail. And that's the same here. You want to get the nutrition into the body, but you don't want to get toxins or unwanted parts of which are in the lumen. You don't want to get them into the body. And therefore, we have a sort of defense system, which is called the intestinal barrier. One of the important things is that the mucosa cells are tightly attached to each other by tight junction complexes. And if these tight junctions work less efficient, then you can see that the distances between the cells will become a little bit bigger, and some substances which normally are not allowed to pass can pass between the cells. And that's what sometimes is called leaky gut, but I think it's a little bit too extreme term to use it. But some leakage can occur, and that will activate this barrier system underneath, activate a lot of cells, and if that is of prolonged character, it can also induce inflammation, because these prolonged immune responses will cause inflammation. Next slide. So examples of aberrations of intestinal barrier function in just normal practice are, for example, inflammatory bowel disease, but also irritable bowel syndrome. Especially subjects with diarrhea predominant irritable bowel syndrome suffer from changes in intestinal barrier function. Metabolic syndrome overall is associated with that. In some degree also aging, but probably more related to the diseases which occur during aging. Prolonged stress, either physical or psychological stress, can induce changes in intestinal barrier function. Strenuous exercises are very well known, dehydration as we have shown, and even we think that in neurodegenerative and in other neuroinflammatory disorders there can be some changes in aberration of intestinal barrier function, and perhaps they even can be present before symptoms of neurodegenerative and neuroinflammatory diseases occur. So it's quite common, and therefore it's also very important. Next slide. So we think that if you have the right diet and the right microbiome, and that is producing, for example, a sufficient amount of short-chain fatty acids like butyrate, you have a certain extra resilience against damage of your barrier function. And that's what you see in the middle panel of these three parts of the slide, where there is a disturbance of barrier function, but it is actually counteracted by, for example, the effect of short-chain fatty acids and other beneficial parts of the gut mucosa and the intraluminal content. However, if you are also devoid of these short-chain fatty acids and these other molecules, then you see actually that the gut mucosa is ruptured, and that will really affect the barrier function and will have a sort of, start immune responses and inflammation. Next slide. So if we try to combine this together, how can then the microbiota intestinal barrier affect brain function? Well, what we see is that what I already explained is that impaired intestinal barrier function causes this local, but after all also a systemic immune response. It causes mast cell degranulation, and mast cells produce, for example, proteases and histamine, which actually also negatively affect the barrier function itself. So you get a negative vicious circle, you get neuroinflammation, you get changes in the enteric nervous system, and that all results in a change in vagal nerve activation from the enteric nervous system to the gut. So the local inflammation and immune responses in the gut will change actually the activation of the brain. And that is the most important way how this kind of barrier function changes and enteric nervous system activations change the brain. On the other hand, also we see changes in the matrix of the enteric nervous system by the glia cells, and they are important to actually change the signaling of the brain to the gut into signals of the enteric nervous system, and that can also go somewhat wrong if you have prolonged activation of the immune system in the gut. Next slide. So this is the most complicated slide, and I will not tell you everything about that slide, but just guide you through the most important. Because this slide shows how changes in gut barrier affect the brain. What you see is that, and how it actually uses the serotonergic signaling for that. So we come back to one of our first slides about serotonin and tryptophan. And what you see here is that if the gut barrier is impaired, for example, by transfer of lipoprotein saccharides, this will affect inflammatory responses. And by this inflammation and immune responses, what you will see is that you will induce an enzyme, which is called IDO, and that enzyme actually uses tryptophan into other routes. It will actually produce other molecules than serotonin. So what you see in an activation of inflammation locally in the gut, as well as systemic, which is also reflected in the liver, is that less tryptophan will be available passing the blood-brain barrier to the brain, because it is used by these IDO enzymes and other enzymes into other routes than serotonin. So the tryptophan is heavily scavenged in the system and can't reach the brain in the same level as before, and that actually reduces serotonin production and activity in the brain, and that has directly a negative effect on emotions, can cause depression, anxiety, changes in appetite, and so on and so forth. Also, it can also relate it because this induction of this IDO enzyme also gives local neuroinflammation in the brain, and that actually makes the whole situation worse. So this is the reason why in inflammatory disorders there is less tryptophan available in the brain to produce serotonin. And that's also the reason why inflammatory diseases like inflammatory bowel disease, chronic arthritis, and so on, a metabolic syndrome, are associated with, for example, depression and changes in cognition. Next slide. We have actually looked at the effect of butyrate more on this on the neurosystem, and actually seen that in cases of challenges of nerve endings, in this case simulated by fibroblasts, by, for example, oxidative stress, that butyrate can actually protect these neurons and fibroblasts from oxidative stress. So we think that butyrate can protect the enteric nervous system, and small amounts of butyrate will also enter the brain, and perhaps, but we can't prove that yet, perhaps can also protect the brain against oxidative stress locally. And this is done by providing and protecting tryptophan transport over the membrane. So you still be able to produce your serotonin because of the butyrate. Next slide. So some clinical examples of how diet, microbes, barrier function, inflammation, serotonin, stress, and mental health interrelate. Irritable bowel syndrome is associated, for example, with depression and anxiety. About 30–40% of irritable bowel patients suffer from this. But if you look at subjects with diarrhea, predominant irritable bowel syndrome, in my practice, it's much higher. It's over 90% of these patients who have either qualify for depression or qualify for an anxiety problem. And we also see that a lot of these irritable bowel patients with diarrhea, about 60%, suffer from changes in the intestinal barrier function. So here we have a lot of challenges, and we are not that far with nutritional treatment of irritable bowel syndrome that we can change all these negative changes in IBS, but we are on our way. The other example is inflammatory bowel disease, where also more of 40% of subjects actually at the same time suffer from symptoms of depression or anxiety. And this is directly related to the inflammation in the gut and the systemic inflammation. So ways of treating inflammation could be, of course, by medicines, but we also know that substitution with omega-3 fatty acids can have an effect on the immune system and on inflammation. And this is used in inflammatory bowel disease in some countries. It's not really practiced in, for example, irritable bowel syndrome yet. But also you could look at probiotics and prebiotics, which can change the barrier function, for example, and we have several proofs of that in the case of both prebiotics as well as probiotics. So there are a lot of ways to do that. The third example I mentioned is Parkinson's disease, and the reason for that is that it's more prevalent than you think. It's estimated that about 8 to 9 million people in the world suffer from Parkinson's disease, and it is actually increasing. And we and other researchers and clinicians think that in a part of Parkinson's disease patients, Parkinson's disease starts in the gut. And a lot of Parkinson's patients actually have experienced sometimes lighter, sometimes more severe gastrointestinal problems about 10 years before the onset of symptoms of Parkinson's disease. So we think this is a gut-brain signaling issue, and we are now heavily investing on research on that, how we can prove that, how we can find the mode of action, how it works, and next, how we can intervene there with nutritional measures. Next slide. So we have talked about parts of the diet like prebiotics, probiotics, and so on. But of course, the diet as a whole has the importance. And how can the diet affect the gut-brain axis? Next slide. Because it's the diet does not only contain prebiotic agents or probiotic agents, it contains antioxidants, it contains different fatty acids and so on. Well, we do not exactly know how it works. For example, some of the problems is that we still not know whether a lot of these effects on nutrition, on gut-brain interaction is on the small bowel level or on the colon level. Probably it's much more on the small bowel level than we think. Is it an interaction with just the gut microbiota, or is it a direct interaction of nutrition with the brain, like we know from, for example, Red Bull or alcohol? It has a direct effect on the brain. Or is it a combination of these things? And if we relate it to the gut microbes, is it a direct effect of these microbes on the gut system and the whole body, like, for example, with probiotics? Or is it more by the compounds they produce, like the prebiotics and the postbiotics? And what are these compounds? I've mentioned many compounds important in gut-brain signaling. And what are these compounds which are the most important one? And how important is it to enforce and strengthen intestinal barrier function by nutritional measures in order to beneficially affect gut-brain interactions? And how important is it to act anti-inflammatory? And we also now ourselves perform studies about using anti-inflammatory diet and try to find out how it affects the gut-brain interaction and gut and mental health as well, of course. And how much is it important to improve immune function by nutrition? So it's still a very complicated field, but step by step we come forward. Next slide. So if we look at what could be the functional applications in the end, I think that we will be in some years from now be able to, by nutritional means, attenuate stress impact on the gut function. And in my last slides of this presentation, in a minute or so, I will show you an example of that. I think that we can have a beneficial effect on depression and anxiety. And mind you, mental health and disturbance of mental health is the new epidemic in the world, especially in the Western world. So we are not moving from the epidemic of obesity, it's still there, but we are actually moving to another epidemic, and that's the epidemic of decreased mental health. Of course, we think that you can modulate satiety by different ways, but perhaps even we can prevent or delay neurodegenerative or neuroinflammatory disorders, to start with Parkinson's, but why not later on multiple sclerosis or even Alzheimer's disease? But the big challenge is still we need to personalize, because there is not one nutritional intervention which is best for each person. So that is really the big challenge for the nutritional field, and it also is the same sort of challenge if you relate it to gut-brain interactions. Next slide. So to finish off with just an example of how you can move forward into personalized nutrition and try to personalize your dietary changes and dietary interventions. Here I show you a model where we use a surrogate marker of mental health. So it's not a mental health as such, but it is a prediction of parts of mental health by looking at brain activity using functional magnetic resonance imaging, fMRI called. And you see here that we stress the person in the camera with what we call a EAT paradigm, EAT paradigm. And what you see here is that you see different expressions on the right lower panel, fear or anxiety or anger or whatever. And the person in the camera has to see what matches with on the lower panel with the upper panel. So in this case, you see anger in the upper and in the left one. So you have to push a button that it is the left one. So you are emotionally engaged, and these shapes and these faces are not that very clear in their emotions. So you have really to engage. And the control situation is exactly the same sort of procedure, but now you don't see any emotions. You see such a circle or an oval, and you have to show what is matching. So that is actually subtracted from the other one. And then you can see what actually the emotional activation produces for signaling in the brain. And then you can intervene with nutritional interventions like we did with probiotics. Next slide, and I can give you some examples of the results of that. In this panel or this slide, you see actually highlighted some parts of the brain which are differently affected by this emotional. They are all many more parts of the brain are activated by emotion, but these parts are differently affected in case if you take a certain probiotic during 4 weeks. And mind you, these were totally healthy subjects. And you have to correct for multiplicity. So this is quite solid statistical evidence that some parts of the brain are affected by probiotics in handling, especially emotions. Next slide. We also could show on a high statistical level that there were even small changes in terms of anatomy, structural changes in the brain. And all these changes which we have seen, they all go into the same direction, showing that it seems that this certain probiotic mixture had an effect that the brain needs less energy to cope and handle a certain acute stressor, which could be a positive effect. So it's what we call a surrogate marker of mental health. It's of course not the proof, but at least it shows which kind of subjects respond and which kind of subjects not respond, as well as you get an insight on the mode of action. Next slide. So to summarize my presentation, the importance of diet for mental health is by the gut ecosystem and by changing the microbiome. It has also a direct effect on gut and brain function. We have previously shown that diet, especially the fatty acids in your diet, can change the brain's composition. We think that diet is important for keeping up intestinal barrier and immune function, that it can affect beneficially inflammation locally as well as on a systemic level. And of course, it can also stimulate the production of certain bioactive compounds in the gut, which can signal to the brain as well. Next slide. And by that, I want to finish and thank my team for all the research done. And if you are more interested in our research, you can also, next slide, look at our Twitter and Instagram account. Twitter is called X, I think, nowadays. Thank you.